Good morning and afternoon. Thank you all for taking time out of your busy show schedule today to come to today's webinar, Applications of Advanced Genomic Tools in Aquaculture Breeding Programs. My name is Jason Hine with LGC Biosearch Technologies, and it's my pleasure to be your moderator today. Just a few housekeeping items before we get started this morning. We'll be taking questions at the end of the webinar in the form of a panel discussion. To submit a question, type it into the question chat box and we will collect the questions and then answer them as we have time at the end of the webinar in the form of a panel discussion. Any questions we don't have time to answer, we will answer via email following the webinar, when we also provide a link to the recorded presentation from today. At LGC, our mission is science for a safer world, and we achieve this through collaborative partnerships with our customers, providing genomic tools that enable our customers to overcome the sustainability challenges we all face in the agriculture space. Each of our speakers today will share with you how they have answered this call. In each presentation, the speakers will share with you not only excellent science, but also how the science is helping drive the advancement of aquaculture species to secure our global food supply as the world faces diminishing resources. Our first speaker today is Dr. Marine Herlin from Coolmerex, a part of the global aquaculture company Cook Aqua. She completed her PhD in aquaculture in Scotland, where she focused on broodstock management of Atlantic cod farm populations using DNA microsatellites for pedigree reconstruction. After completion of her PhD, she moved to Spain, where she managed the bass and sea bream hatchery of Coolmerex, now part of Cook Aqua. Currently, Marine holds the title of Global Genetics Manager, overseeing and managing the breeding programs for Cook Aqua, including sea bass, sea bream, and Atlantic salmon breeding programs. Dr. Jose Yanez is our second speaker, who is the Director of Research and Development and Faculty of Veterinary and Animal Sciences at the University of Chile. Key to his research is determining the genetic architecture of complex traits, which he then takes forward into the design and implementation of genetic improvement programs in fish populations, as well as genome-wide association studies and genomic selection in aquaculture species. Our third and final speaker is Dr. Mitchell Lucas, Director of Genetics at American Pinnade. Dr. Lucas graduated from Fresno State in California, University of California, Riverside. Prior to American Pinnade, Mitch was using DNA markers and CASP genotyping technology for selective breeding of new varieties of bell peppers at Syngenta. When Mitch joined American Pinnade in 2019, he implemented the technology to advance the breeding program and deliver improved growth and survival rates of seed stocks for shrimp farmers around the globe. Thank you in advance to all of our distinguished speakers for sharing your expertise with us today. Marine, I'll hand over the presentation to you and you can start us off.
So for the next 15 minutes or so, I will share with you our experience at Cook Aqua in developing and applying tailor-made genomic tools in our commercial Atlantic salmon breeding program. So to go quickly through the presentation, first I will give you a brief introduction on our story at Cook Aqua for our breeding program in Eastern Canada. Then I will move on and show you some of our main results in terms of growth, genetic gain, using our traditional block model. And then I will speak a little bit about the development of a new molecular toolbox. We've been working very hard over the last 5 years to do so. And then I will carry on and show you some applications and how we are implementing those tools in our breeding program. And I will finish by giving you some key areas that we have on the agenda to further develop. So to start with, Cook Aquaculture has been managing its own breeding program for Atlantic salmon St. John aquaculture strain since 1998, so it's over 20 years now. The breeding program has been operated since the beginning at Oak Bay Hatchery, which is a hatchery located in New Brunswick. It's a freshwater land-based facility with a production capacity of 30 million eggs per year. And traditionally, we were rearing the families communally and the future broodstock candidates were being selected at 3 years old, based mainly on their growth. And we were using also SIB groups, which were reared at sea, to get some complementary data to evaluate growth and maturity. Then we also started to work on a project in 2014 with the collaboration of the University of Guelph and funding from both Genome Canada and Genome Atlantic to develop advanced genomic tools. So that's how we embarked in the genomic area. And in 2015, there was some building being put up to set up a 100-tank individual family rearing unit at the hatchery. The same year, we had the first version of our high-density SNP chip for our strain. And the following year, in 2016, we started to compute genomic breeding values for growth and lice resistance, and we applied those values in our breeding program to select our future broodstock. Moving on to 2018, we had a new version, an improved version of our high-density SNP chip array, which was developed together with the Norwegian company Aquagen. And in 2019, that's where we started to work with LGC Biosearch Technologies to develop a cost-effective low-density SNP panel to be able to further apply our SNPs in our breeding program. And we also had in 2019 the construction of a new building where we have now the capacity to rear 250 families. And we are still working on our panel together with LGC, and we are validating our SNPs for parentage analysis and continent of origin assignment. So to give you a little bit of background on our work, we started to work on growth improvement in our strain, as I said, 20 years ago. So our broodstock candidates are evaluated at 2 and 3 years old, and we record individual phenotypes for body weight, length, gender, and maturity. The phenotypes and genotypes are also collected from SIB groups, which are reared at sea, about 2,000 to 3,000 individuals per year class. And the pedigrees are reconstructed using the genotype information from a panel of 245 SNP markers. We've been estimating breeding values using a traditional BLUP animal model. And we've been developing a model together with the University of Guelph to include 8 traits. And we are taking into account a gender dimorphism we found in our strain for growth. And I give you the reference to the 2 publications we had in 2018 on this topic. So with this model, we calculated heritabilities for growth at sea. And over the last 12 years, we estimated we gained about 0.9 kilos for males and 0.8 kilos for females at harvest size. So which is fairly significant. Our harvest size is 5 kilos. So about a kilo is fairly significant. And as you see in the following graph, you see the evolution of this gain. So we are working with 4 different year classes, and we basically select on each year class. And you see the last year we made a big jump in our gain on growth, and that's also because we've been including genomic selection. When we look at commercial data and we compare spring entries at sea between 2013 and 2016, we realized that actually we have genetic gains, which is effectively transmitted to the commercial production. So that's also very good news, because we managed to get some good indicators that our breeding program is working in the right direction. And that gave us basically the baseline to start working on developing genomic tools. So for that, we started to develop together with SciGene, Aquagen, and the Norwegian University of Life Sciences an array, a high density SNP array. So we've got two versions, one which is 50K and the other one which is 62K. And we've been working on a third version using the genome reference for our strain. So since 2014, we have genotyped about 16,000 animals, and they are from our broodstock candidates, of course, but also saltwater evaluation and disease resistance animals. And since 2016, the company has selected candidates based on growth, bacterial and bacterial kidney disease and sea lice resistance using the genomic breeding values we've been computing. We also used the genomic data to run some GWAS analysis, and we've been using, for the moment, the European Atlantic Salmon Genome Assembly. So we are on the way to review those GWAS analyses using the new reference genome for our strain, but we already identified a number of SNPs which have significant effects on the phenotypes we are recording, and we are planning to use this information probably to do marker-assisted selection. And moving on a little bit on the genomic tools, we have worked, as I said before, with LGC Biosearch Technologies in developing a low-density SNP panel. And we worked on the first version in 2019, including 632 SNP markers, which were specifically selected for parentage analysis and continent of origin, and also for gender assignment. And we make good use of the high throughput, cost-effective SNP genotyping technology that LGC gave us access to, and we've been using the information to apply in our breeding program. So the first genotyping project was run, as I said, between April and June 2019. We genotyped about 4,000 candidates with this tool. And we've been refining this list of markers. And now this year, we've been validating a panel of 400 markers that we are further working on to refine and be able to select our broodstock candidates. So for us, the SNP technology has been very flexible and very reliable, and it offered the possibility to include new informative SNP markers every year. And we are planning, as I said before, to implement marker-assisted selection. And the fact that it's very cost-effective and very flexible, it allows us to genotype a large number of animals. So not only broodstock candidates, but also animals that have been phenotyped for different traits based on data from our commercial operations, or also, as I said, disease challenges that we are running in laboratories. So moving on and talking to you about the integration of those tools into our breeding program, we've been successfully developing a parentage analysis assay, which consists of 243 SNP markers, and they are very reliable, and we managed to assign successfully close to 98% of our offspring to a single family. And we are also using this tool to assess the continent of origin. So that's a requirement for Canada. For us, it's important to show that the fish that we are breeding are coming from a local strain. So for that, we are using a list of 55 SNP markers, and with the allele frequencies that are different between the EU and the North American stocks, we are able to assign the continent of origin of our fish and show the authorities that we are effectively breeding wild origin North American Atlantic salmon. We have also a tool for gender assignment, which is very accurate, 98% accuracy, and it consists of 6 SNP markers. And now, as I said, moving on to the high-density SNP chip, we have been computing GEBVs for growth, lice, and BKD resistance. And in 2019, we have developed a single-step deep loop analysis to be able to combine the pedigree and the molecular data we have on all our fish. And we also produced the first heritabilities for lice and BKD resistance, which are two diseases that are very important in farming salmon. And we saw that we can effectively select for those two diseases, and also that they are not genetically linked. And we are using also the genomic breeding values to effectively select the best individuals from the best families we have to produce the crosses for the next generation. And effectively, since 2016, the parents used to produce the select crosses are among the best tips from the best families. And in terms of further developments, we have plans to add large effect pathogen SNP markers to the sex SNP assay we have with the collaboration with Biosearch Technologies. We also look at combining genomic selection for the breeding nucleus and marker-assisted selection for the commercial production of juveniles. And we are also looking into developing cost-effective CASP assay for parentage analysis and continent of origin assignment, again, in collaboration with Biosearch Technologies. And to conclude, I would say that the introduction of genomic tools in our breeding program has significantly improved selection accuracy for both saltwater growth and disease resistance in our stock. And thanks to the sex-SNP technology, we have managed to screen and evaluate more broodstock candidates used in our commercial egg production lines. And over the past 2 years, we also developed a very strong service provider-customer relationship with Biosearch Technologies. And we are looking now into developing new tools for our bass and bream operations in the Mediterranean, so the Climatic group. And thank you very much. And if you have questions, I'm really happy to answer them later. Thank you.
Thank you, Dr. Herlin, for that wonderful presentation. Next, we'll hear from Dr. Jose Yanez, Director of Research and Development and Faculty of Veterinary and Animal Sciences at University of Chile.
Today, I'm going to present the application of low density SNP panels to implement or for the practical application of low-cost genomic selection. I would like to thank to LGC and Biosearch group to invite me here to present part of the results that we have obtained using these technologies. So part of the results that I'm going to present here are, some of them are published in details in these two review papers. One of them in the journal Reviews in Aquaculture, which is mainly focused on the advances in genetic improvement, including genomic selection in salmon and trout species from a Chilean perspective. And the second one is focused only in tilapia, is published in Animal Genetics, and it shows the application or the early developments in the applications of genomics tool to accelerate the genetic improvement in tilapia. So in general, the breeding objectives of breeding programs in salmon and tilapia include traits that are difficult to measure in the selection candidates. And basically, these traits are included if we're using conventional pedigree lab methods or approaches, are included mainly based on family information. So this approach has the disadvantage that the accuracies of the estimated breeding values are low, And also there is a trade-off between the selection intensity and inbreeding, because we're only exploiting between family and not within family information. So as a consequence, this can lead to limited genetic progress for the breeding of this species and this particular trait. But genomic selection, which is an approach that incorporates pedigree data, phenotypes, and also SNP information, can increase the accuracies of estimated breeding values, and with this increase the response to selection and also accelerate the genetic progress. But one drawback of this approach, the genomic selection approach, especially for low or small producers or small aquaculture producers, is that genotyping represents an extra cost, and it's generally not a marginal extra cost. So genotyping, in general is considered as a limiting factor for the practical implementation of genomic selection in aquaculture species. How does genomic predictions or genomic selection works? The basics behind the genomic selection is very simple. We have here a reference or a training population in which we measure phenotypes, and also we have genotypes for these animals. Let's say disease-resistant trait or trait that we measure at the processing plant, like quality of carcass or fillet yield or color in the case of salmon and trout. We have these animals that with phenotypes and genotypes data, and we train the models or the prediction equations with this data in order to predict the genetic merit of animals that are the selection candidates, which only have genotypes. We don't have phenotypes for these animals. Let's say a disease-resistant trait. We don't want these animals to be exposed to the pathogens. So we basically predict the genetic merit of these animals based on the models already trained with genotypes and phenotypes in highly related animals from the selection candidates. For instance, full SIPs or half SIPs from these selection candidates. Then with these predictions, we make selection decisions, and we select the breeders for the next generations using genomic breeding values with high accuracy or higher accuracy than the conventional pedigree genetic evaluation. So we have here a way or an approach to decrease the cost of genomic predictions. As I mentioned before, the genotyping is an extra cost that is considered as a limiting factor for the practical implementation of this approach in aquaculture breeding programs. And the imputation is basically the prediction of the missing genotypes using information from a reference population. So we can go from low density SNP panels that are cheaper than higher density ones and impute or predict the denser genotypes. So we have a reference population with all of the genotypes available for this population, and we have a target population in which we have some missing values or missing genotypes, and we can impute or we can guess from the reference to the target population the missing genotypes. So we can use this approach to use low density panels that are cheaper compared to high density one in order to decrease the cost of genotyping. So this is an example in Atlantic salmon. We used different scenarios and different combinations of low density panels. For instance, using all the sires, dams, and a proportion of the full SIPs from the offspring, or even using only sires, no dams, and only a proportion of the sibs. No sires, only the dams and a proportion of the sibs, and no parents at all, only a proportion, let's say 10% of the siblings, and only the parents without using siblings of the offspring. So these are the numbers. All of the details of this paper are reported in this of this work are reported in this particular paper in Aquaculture Journal. We have here 3 different densities of low density SNP panels: 0.K, that means 500 SNPs, 3K, and 6K. And you can see that the accuracies are always above 80%. We use here true genotypes in order to estimate the imputation accuracy, and it was always higher than 0.8. So that means that we can impute with high accuracy from low density panels to high density panels, even from 0.5K to 50K, which was the high density panel used here. So low density SNP panels can be used to accurately impute to higher densities, and we can use 2 technologies provided by LGC, Biosearch Technologies, like CASP, which is a PCR-based genotyping approach, or SEC-SNP, which is a targeted GBS approach. That can be good options to genotype these low density panels with a decreased cost. And we were also interested in evaluate the impact of this of the use of imputed genotypes on genomic predictions. And as you can see here, we have on this blue bar here, the first bar is the accuracy of breeding values using only pedigree blab, and then we have the accuracy of breeding values or genomic breeding values using a 50K SNP array. And then you can see that there is no decrease in breeding values or the accuracy of breeding values when we use imputed genotypes, but only for the 0.5K scenario or SNP panel, which is kind of marginal, the decrease in the genomic selection accuracy. So that means that for the practical implementation of genomic selection, low density panels are appropriate to impute to higher density and to predict the genetic merit of the breeders. So we did something similar for tilapia, and we went here from 0.5 to 3K and also 1K to 32K SNP panel. As you can see here, these are the prediction, the imputation accuracies here are, all of them on average are above 0.8. And you can also see that there is kind of variation across the genome in the different chromosomes. There are some regions that present lower imputation accuracy, but in general, we can also say that the imputation from 0.5K or 1 or 3K until 32K in tilapia are also accurate. So this is something that we can exploit and we can use to decrease the cost for tilapia genomic selection. The same as in Atlantic salmon, here we also tested the impact or evaluated the impact of imputed genotypes on genomic predictions. And as you can see here in the left hand side, you can see the use of the true genotypes, 32K in green, 3K in red, 1K in yellow, and 0.5K in light green. If we use true genotypes, only true genotypes, the accuracy of genomic predictions decrease, right? Decrease in this way. But if we use imputed genotype, the decrease of on genomic predictions is lower. So we can also use in tilapia low density panels, let's say 1K SNP array, to impute to higher density and decrease the cost of genotyping. So going to the cost and to the this is something that maybe is of interest of the producers. We have a base case scenario, which is the scenario A, on which all the animals are genotyped using a 50K SNP array. We also have a scenario B, which all the parents and a percentage of this offspring are genotyped using a high density 50K SNP array, and the rest of the animals are genotyped using 6K SNPs. The scenario B, C and D are similar to scenario B, but the low density SNP panel is a 3K in the case of scenario C and 0.5K in the case of scenario D. So the cost of genotyping, we assumed $50 for a 50K SNP array, $25 for a 6K SNP chip, SNP panel, $20 for 3K SNPs, and $10 for 0.5K SNPs. So when we consider the scenario A as the base case scenario, and the other three, B, C, and D scenario across breeding population of different size, I mean, that means the size or the number of animals to be genotyped from 4,000 to 10,000. We can even reach a decreasing cost between minus 70% or even marginal when we use 6K. It's important to note that we assumed here a decrease in 10% of the cost of the high density genotypes when we increased 1K SNP in the genotyping of the high density SNP panel. So as an example here, we have a genome-wide association study. This is a practical example from a genome-wide association study for growth at the extreme temperature in trout. We genotype 192 fish with a 20K sex SNP panel from LGC. Other set of animals, about 600 animal or more than 600 animals, were genotyped with 1K sex SNP panel, and we imputed from 1K to 20K to perform GWAS for 80 average daily gain at 7 degrees in trout, and also average daily gain at 22 degrees in a breeding population of rainbow trout. So when we ran the GWAS, the genome-wide association study, we detected two peaks. One peak for average daily gain at 22 Celsius degrees. It's a gene called ATAT2B, which is related with thermal stress in different aquaculture species or fish species like catfish, bluefin tuna, Arctic char, and also longjaw mudsucker. So this is an interesting candidate gene. It's only significant at the chromosome level. It's located in chromosome 8. But when we ran the GWAS for average daily gain at 7 degrees, we had surprisingly hit or a significant region which is genome-wide significant for a gene called IRF1, which mediates the cell growth inhibition, and also it's also modulated by the growth hormone. So it's a candidate gene or very important candidate gene for this particular trait. And one of the SNPs is present in one exon of this particular gene. So we have a couple of interesting candidates or putative genes. So in terms of the breeding, when we want to maximize the accuracy of genomic predictions, we can go for QTNs. That means the causative mutations that are affecting a particular trait, which has been proven that for these QTNs, we can, if we use them to predict the genetic merit, instead of using whole genome sequences or even high density SNP markers, we can maximize the prediction accuracy for estimation of or prediction of the genetic merit of a particular trait. As you can see here, if we use causal SNPs in the last row of this table, if you use causal SNPs and we have two traits, one controlled by 20 QTNs and another one controlled by 100 QTNs, and if we use or we only incorporate these 20 or 100 QTNs in the genetic evaluation for these two particular traits, we can get 0.98 and 0.95 of accuracy using only 20 or 100 variants. So this is even higher than using sequence variants, which is marginally better than using high-density SNP arrays and also medium-density SNP arrays. RAD-seq, it can be even worse than only pedigree blab, depending on the coverage and also depending on the distribution of the markers across the genome, and also on the informativeness of the SNPs. Based on this particular study, simulation study from 2015, we used prioritization of SNPs for genomic predictions, in which we complement the analysis showed before. We had 192 fish genotype with a 20K sex SNP panel and plus 614 fish genotype with a 1K sex SNP panel, and we imputed from 1K to 20K. Then we perform whole genome sequencing of on breeders of these animals. So we sequenced more than 100 rainbow trout parents from this breeding population using a coverage of 10x. We discovered 22 million of SNPs after running an algorithm to discover variants. We measured body weight after 2 months of thermal stress, above 20 degrees Celsius. We used 1.8K animals with phenotypes and 800 animals genotyped. We imputed up to 1.4 million of SNPs with high accuracy, and then we used weighted single-step GBLAB to perform a genome-wide association study and prioritize SNPs based on the genetic variants explained in order to select or pre-select SNPs for genetic evaluation. So we estimated the accuracies of genomic predictions using pBLAB. That means no SNPs were used for this scenario. We also use 50K SNP panel, choosing a 50K SNP from a random or randomly across the genome. We also used whole genome sequence. That means all of the 1.4 million SNPs available. And at the end, we used 20K SNPs that were prioritized based on the variance explained in the weight single-step GBLAB GWAS, and they were chosen in descending order. That means the most important SNPs, or the SNP that explain the highest or the higher percentage of the variance, were chosen for this 22K SNP panel. And as you can see here, there is an increase in the accuracy when we go from PBLAB to 50K randomly chosen markers of about 5.6%. Then when we used whole genome sequences, it was only 1% better, or even less than 1% better than 50K randomly chosen SNPs. So there's no improvement when using whole genome sequences. But when we use prioritized the SNPs, we reach a maximum accuracy that was 22.5% better than the PBLAB. So we were able to increase the accuracy by using or choosing or prioritizing SNPs based on the importance assessed by genome-wide association studies. So in terms of the conclusions, we can say that genomic selection can accelerate the genetic progress for disease resistance and other important traits in aquaculture species. The imputation from low-density SNP panels, for example, CASP or sex-SNP, to high-density genotypes decreases the cost of genomic selection in aquaculture down or even up to 80% of the original cost of using dense information by the use of 50K or even higher density SNP panels or SNP arrays. Weighted single-step GBLAB and ultra-dense SNP information from whole genome sequences can be used to increase genomic prediction accuracies by means of SNP prioritization. So choosing SNPs based on the genetic variance explained instead of using randomly or even use whole genome sequences can be or can generate better accuracies for genomic predictions, depending on the trait, of course. So I would like to invite you, all of you, to this International Symposium of Genetics in Aquaculture to be held in Puerto Varas in Chile in November, between November and December of this year. These are the social media links in Facebook, Twitter, and Instagram. And also you can check more information on the website of this event here. And I would also like to thank all of the collaborators for their input and also funding this research. Thank you.
Thank you, Dr. Yanez. To be sure that we have enough time for our final speaker, we'll move quickly to Dr. Lucas, Director of Genetics at American Pinnade. Dr. Lucas, the floor is yours.
Okay, hi, I'm Mitch, and I'll present our work on using CASP SNP genotyping to breed better shrimp. I'm with American Pinnade. We're a food shrimp and seed stock business in Southwest Florida. And this is about LGC and CASP, so I wanted to talk a little bit more about how I've used CASP in the past. So it first started in graduate school at UC Riverside. You can see those brown beans up in the top left corner. Those were collected from or developed in southeastern Africa, and we identified 2 QTLs and used CASP to back cross those into a black-eyed type, seen on the right for California. And the seeds we got were a lot larger. So we were tracking these 2 QTLs that impacted seed size. It's kind of interesting to see that you can breed a small seeded variety with another relatively small seed variety and get really big differences in seed size. So that was in grad school, and then I was hired by Syngenta as a vegetable breeder, and I was working on bell peppers. And in the commercial seed industry, CASP is the workhorse. Most serious global companies are investing in SNP genotyping, and I was using that for virus resistance. So breeding new varieties of bell peppers with resistance to some viruses. About 2 years ago, I was approached with the opportunity to lead the genetics program for an emerging player in the food, shrimp, and seed stock business. So that was American Penaid. And our focus there is on survival. And so we basically started with no genetics lab. So we built a lab on the farm, and I got to hire my own team and breed largely by my design, which is guided by CASP. So the next slide. Switching from plants to animals was not that difficult. Aquaculture is an industry of growing importance where species have only been recently bred in captivity. There are many newly farmed species and those that have large spawns. Production environments vary dramatically and would benefit from customized genetic programs. So you can see those 2 pictures there. One are shrimp ponds in Indonesia. Completely outdoor, relatively low input, little control. And then on the right is a picture of a facility in Europe. It's completely indoor using the RAS style of aquaculture. So these are situations where investment in modern breeding can make a big impact. You really need different animals for these environments. And that's essentially our path at API. So there's a picture of our farm from a few years ago. Originally, the company was only in the farming business, helping to address gaps in U.S. access to quality food shrimp. And our Sun Shrimp brand represents that continuing effort. However, we experienced wavering seed stock qualities. It jeopardized our business and much of the global industry. And we began despite the farmer's best management efforts, seed stock genetics weren't good enough. We began developing our own seed stocks, and within 10 years are the leading broodstock supplier in the world for P. vannamei, the primarily grown species of shrimp. That means when you eat shrimp, there's a good chance that its genetics originated from our farm. Our animals are different. Everyone sees it. I can show you genetically. Our farm and broodstock managers can show you physically. Our animal is unique, and shrimp farmers buy American Penaid genetics because our animals survive the reality of shrimp production. Other companies offer sports cars that will grow real fast on a perfectly prepped racetrack. We offer a 4-wheel drive truck that tolerates the challenges of aquaculture. Our germplasm has a desirable chassis that we're continuing to improve. And so really it's the ability of our animals to survive that is a big part of the value of our company and kind of our legacy to build on and maintain. And definitely one of my priorities as a geneticist. So in that effort, we built an on-farm genetics lab. Our lab's a factory of high-throughput DNA extraction and SNP genotyping and data analysis. Here's a picture of the liquid handler that we use for DNA extraction. It's really easy in shrimp to extract DNA in other marine species. It takes us about 15 minutes per sample, but then we multiplex hundreds of samples together. Our normal workflow is 1 day for sample inventory, another day for running the PCR and clustering the results, and then a day for analyzing to support breeding decisions. Here's some photos of the equipment we purchased from LGC to implement CASP in-house. So the Nexar on the left, it combines the DNA with PCR ingredients. It automatically seals the reaction wells and keeps track of samples from barcoding. The hydrocycler there in the middle performs the thermocycling needed for PCR, and the last equipment on the right, the Arraea, scans the completed reactions to measure fluorescence intensities. We chose CASP, this setup, because it's ideal for commercial breeding programs. This has been and continues to be the workhorse employed by breeders in other industries. It's essentially off the shelf, it's repeatable, it's fast and easy to analyze, and it's very tolerant of sample variation and low quality DNA. There are other technologies like next generation sequencing and high density chips, but in my experience, these are mostly used for special projects and not the routine solution. It took us only about 2 months to set up our lab, from the initial meetings with LGC to actionable data. One scientist really runs the entire lab with essentially no error and can process thousands of samples a day against a panel of genome-wide SNPs that we're using. The equipment is well built and hasn't broken down on us, so that's been really nice. There's a lot of benefits to having an on-farm genetics lab. Sample quality is excellent. Turnaround time from collection to analysis couldn't really be faster. And resampling is not a big deal, and we don't have to ship samples to a third party. This means we can use cheaper plastics for collection. We don't need to hire a specialist to manage all the complexities of dealing with a third party. And maybe the most obvious one is that you're independent of another lab's priorities. And when I was outsourcing wet lab work in the past, we had to plan our sampling activities around the other big customers. So in plant breeding, that was soybean and corn. So when you know it's soybean and corn season, don't plan on getting much done with the partners we were using. And I imagine this is even more true now with COVID. The urgency required for COVID testing will likely leapfrog your fish samples and lab priority. So here's a screenshot of the raw data on the left. Like I mentioned earlier, CASP is a robust chemistry that can tolerate some relatively poor DNA. It works by detecting allele-specific fluorophores and PCR products. So in that raw data, the red samples are homozygous for one allele, purple samples are heterozygous, and the blue samples are homozygous for the other allele. The black samples at the bottom of our negative control. And so this is just looking at a population of, I don't know, a few thousand individuals, but only at one particular location in the genome. So then we scale this up and look at many locations depending on the type of population and what the destiny is of those animals. So this slide's about how we've really been using CASP in shrimp breeding. In the last 2 years, we've genotyped over 500,000 unique animals, and we've used the data in a lot of different ways. We collect data on post-larvae, so juveniles, as soon as they're hatched. We also collect it once they're moved. And then we collect DNA again at maturity. And so one of the big things is it's helped us better manage our germplasm and deliver a hybrid breeding program. Using CASP, we're able to manage inbreeding and maximize heterosis and the uniformity of animals. We've also applied it to quality control. We sample every spawn at several time points to validate production integrity and the movement of animals. This relieves any doubts about the batch identity. What is the animal in that particular tank? We're also using CASP to identify and deploy trait-associated markers, so QTLs and and perfect markers. And finally, we've used the tech to compare shrimp originating from different parts of the world, different companies, to understand who has what and where specific genetics are working the best. So I kind of rank these points in terms of, I would say, importance or impact to the breeding programs. Really, the hybrids and quality control, trait-associated markers are still not that mature in shrimp. The competitor analysis is interesting, but it's hard to extract value out of that. So, like I mentioned earlier, our animals are second to none in terms of survival. So I'll focus the rest of this talk on how we use CASP to study survival. CASP allows us to determine genetic relationships between shrimp. It allows me to tell you what family an animal belongs to and quantify family abundance, how much of each family is in the tank. The ability to study multiple families in the same tank is difficult in aquaculture. Historically, families are compared by growing them in isolated environments, more like aquarium type research. But now with the DNA tech, we can study multiple families together in a production environment. So let's look at this table. We've quantified the abundance of 5 families at 2 different time points. The columns labeled PL, that stands for post-larvae. Those are the juveniles, just a couple of weeks old. And then we also sampled them when they reached adult, so over 100 days old. And we used the data to basically determine what family those shrimp belong to in that tank, and how much of them are there at those different time points to calculate the proportions. And it's the change in proportions from which you can deduce relative survival and family performance. And then I guess one of the big learnings we've had is this strong inverse correlation between survival and animal weight. So you can see that they're usually the animals from families that grow the fastest have the poorest relative survival. And this is typical of our work. We've done dozens of these type of survival studies, and you can imagine that we would do this for developing new parental lines. We could also use it for comparing hybrids that are grown in the same tank to understand which ones are best. And so in that chart down there below, it's a distribution graph of the animal weights from those different families. And normally without CASP and the ability to see these different families, you would just most likely go in and take the best shrimp that were the largest. And this work suggests in doing that, you're probably going to compromise the survival. So our approach, which seems to be working, is to select the largest animals from the families with best survival. So prioritizing survival over growth. So it's just a principal component analysis and a structure. That's where the colors came from, structure family assignments. And it's a 3D tool. We can see the relatedness of our different families. We can estimate their genetic distance to breed for hybrids. And it's all based on analyzing the results of the CASP snips. So that is our work on breeding for survival. And so I just want to say thank you to the team. Robin was really the one that believed in us and made a really serious company investment in time and resources to set up our lab. Margaret, Miguel, and Tim for implementing really the breeding program. They're the broodstock, hatchery, and farm managers. And Suzanne Lee, who really runs the daily operations of our lab and also helps with the data analysis. We have shrimp seed stock and brood stocks for sale. We also offer custom genetic programs where we will give you genetic mixes and do a survival study similar to what I showed. So you're developing animals customized to your needs. Finally, thanks to LGC, It's been a pleasure working with them over the years. They really make nice equipment that is user and application oriented. Leanne and Jason from the webinar team, Joe, Mike, and Aaron, and Andres on the sales side. And then when we set up the lab, it was incredible how quick it all happened. And there were several people involved in that and doing some of the background testing and validation work for us. Travis Bruns and the IntelliP support team. So there's a tremendous amount of data that comes from genotyping at the level that we are. And so databasing that and having accessibility and choosing and adding new SNPs to our panel. So thank you, everyone. That's my presentation.
You know, a couple questions that are coming up in the chat box. You know, I think one for everyone. You know, how it seems like there's questions from the community about, you know, how do you choose, you know, your training population? What's kind of the right level of people to choose in your training population? So where do the selection candidates come from, and are they representative of the reference population? And then how do you measure the effect of training population size on the accuracy and cost of your breeding program?
Yep. In general, the aquaculture or fish breeding programs, we used SIP testing. So we take full siblings and half SIPs from the selection candidates to measure disturbance traits. That means disease resistance or carcass quality trait. So the reference population is in generally highly related to the selection candidates. They're siblings, full SIPs and half SIPs.
Maureen, Lucas, you guys want to answer from your guys' perspective about selection of candidates and the effect of a training population size on accuracy and cost?
We are doing exactly the same thing as Jose has mentioned. And for disease resistance, because sometimes we are constrained with the facilities where we can perform the disease challenges, we fit in a subset of the families that we think are the most representative of our training population, and also include, as I said, multiplier and nucleus families. So we also have like the representation of our commercial lines.
Wonderful. Another question for your breeding programs. You know, for someone who's using a high density array today and they want to move to low density genotyping technology and then impute back to that higher density, I guess what are the key things that they would need to have access to be able to make that transition? And then secondly, you know, when you do those low density panels, do you see anything as it relates to family skewedness with that imputation back to a higher density? Dr. Yanez, why don't you start us off, because that came up during your presentation.
So regarding the first question, the things that you don't want to miss when you are performing imputation, to decrease the SNP panel down to, let's say, 500 SNPs, you need biological material from the parents. I mean, you need the parentals. It's good to have the parentals genotype with a higher density. Let's say if you have 120 families or 150 families, you genotype all of the parentals, all of the parents, sire and dams, with a high density. You can even sequence them. It's even better. You will be able to increase the accuracy. I mean, or the number of SNPs to be used in the genomic prediction scheme. So parental information is crucial to do imputation, and also it helps a lot to have some percentage of the offspring, let's say 10% of the offspring genotype with a higher density SNP array. In aquaculture, relatedness and the close relatedness between animals helps a lot for imputation. So if you have the parents, I mean, you have the founders of that. They're not the founders, but they are the population that generates all of the haplotypes in the next generation. So imputation can get very accurate if you have that information, and also if you have all of the parentals genotyped with a higher density SNP array. So it's crucial to have parental information. It's crucial to have an appropriate design. That means the SIP testing, as Marin confirmed that it's very important to have that design. And then the workflow is kind of easy. We have tested in tilapia, trout, and Atlantic salmon, and accuracies are similar. Okay, but one important thing is the design of the low density panel. So you cannot choose any SNP from the higher density. So you need a high informativeness. That means a higher minor allele frequency of the SNPs to be used in the lower density panel, and also distribution. You can take physical distribution across the genome if you have information of physical distribution. But we have seen that even better than physical distribution is linkage disequilibrium. So if you run by linkage disequilibrium, you can get better. You can get a very high informative SNP panel, low density SNP panel, even without having a reference genome. So, but then to impute, you need a reference genome or at least a linkage map to position the markers, right? But there are different ways and different designs depending on the species and the genomic information available.
That was a great explanation. Thank you, Dr. Yanez. Maybe one more question, really on kind of the decisions from high density arrays to maybe low density SNP panels, such as targeted sequencing, and then PCR-based panels, which are even lower density. I guess from all your programs, you were kind of faced with that question of what technology do I use? What were kind of the driving factors for you as you were selecting between targeted sequencing or a high density array or in CASP technologies, as you're trying to figure out which technology makes the most sense in terms of cost, quality, and speed for my particular program?
Sure, I can talk a little bit about that. For us, it takes a lot more expertise to handle next generation sequencing type of data. You need to think about how you're going to store and move that amount of data around. The CASP is really kind of just off the shelf for us. Yeah, and we chose it. That's what a lot of the other industries are using for routine breeding decisions for tracking specific trait-associated markers. They mainly using just a few dozen. And the SNP, the high density arrays and the sequencing can really become important when you're doing the GWAS and making those association studies, maybe doing some type of detailed pedigree analysis. But yeah, we've really benefited from CASP as kind of being routine. It's so quick. The analysis is daily and can be so automated. That's been my experience.
Yes, well, I agree with Mitchell in the way we are processing in our breeding program. We've been relying on high density SNP arrays for getting the detailed information we needed to look for doing the GWAS analysis and look for SNPs that are associated to our traits. And we keep using the high density SNP arrays for a subset of our population to run the GEPVs as well. And we moved to developing a low density panel, moving from SexSNP to now CASP to be able to carry out more routine procedures like parentage analysis and maybe moving forward to marker assisted selection, which allows us to genotype a lot more animals.
Absolutely. No, thank you all for that insight. you know, a lot of great considerations, you know, our people need to be making as they're choosing a technology. I realize we're about 15 minutes over, and I apologize for it going a little bit late. But on behalf of LGC, you know, thank you very much for sharing your time and your expertise with us today, and the excellent science that you guys are doing. I'd also like to thank all the people who stayed on a little bit late for the participation, your attention and thoughtful questions. And if you'd like to learn more about how Biosearch Technologies can assist in advancing your breeding program, I encourage you to reach out to one of our customer service teams with the emails on the screen. For any questions we didn't get to today, we'll be answering those in the follow-up email, which will also have a link to this recording in the presentation from today. Thank you, everyone. Have a fantastic rest of your day. Bye.